So if everybody's up to speed with how a cell membrane is put together, how things go in and out of the cell membrane, those transport mechanisms, the general knowledge about the different organelles that can be found in a cell, both animals and plants, and understanding on what they do in a sentence or two, then you're up to speed. Okay? Now we're going to go and focus our attention on going closer and closer inside the nucleus and talk about that wonderful constitution to the United States, the blueprints that makes all of us human, the DNA molecule. And that's found in the nucleus. I'm going to show you how it's put together. We're going to show you how it replicates in preparation of making going from one cell to two cells, because those two cells have to have the identical information that was in the original cell. So we had to duplicate it. Okay? And that duplication is probably going to be, well, I know it's going to be new for all of you, but this is probably the hardest material that's going to appear on this next exam. So I'm going to go fairly slow with you. I'll do my art therapy up there. I got a little 2-3 minute animation of this, but this is really the ones that are going to separate the A's from the B's, or the A's from C's. So just pay attention over here. The first part is just how the DNA is structured, but then we're going to talk about the DNA replication. So we'll pay more attention to that as we are fine with how it's structured with the bases and stuff. All right, so let's talk about DNA, okay? Genetic material, chromosomes. And I explained to you what chromosomes are. It's the whole encyclopedia set, okay? All the knowledge is put into this encyclopedia set. Now, you can't just have one book, because that book is going to go pretty from the floor to the ceiling. So we just break it up into 46 volumes, or 46 chromosomes, okay? So there's certain information, so not every chromosome has the same information. Not every volume of the book has the same information. Does that make sense? You want to learn about roaches, that's going to be in volume 23. It's not going to be in volume 1. So every chromosome has different information, okay? But the whole thing of the DNA, we call the genome. The genome is knowing where all the information is. It's like the contents of all the information that's in the encyclopedia set. This is on this page, this is on this page, this is on this page, okay? That's what we call a genome. But all this information is on chromosomes, okay? And it's the map of you. We have 46 chromosomes. You got 23 from mom and 23 from dad, okay? So there's 46 chromosomes in every non-sex cell, all right? We call these somatic cells. Not a sex cell like a sperm and an egg. That's different. That only has half. So if you have a sperm with 23 chromosomes, it's half of 46, right? And an egg that has 23 chromosomes, they're going to unite, and then you've got one full cell of 46 chromosomes, okay? We're going to talk about what happens later on should one sperm have two sets, or let's say a full set in there, all right? And then you've got one set over there. You've got 3 sets of a chromosome. Not a good thing. We call it trisomy, and that is the number one reason why there's miscarriages. But we'll talk about that later, okay? Now, there's gametes, and the gametes are the egg and the sperm. All right, there's sex cells. There's only 23, as I mentioned to you. The 2 unite, and that makes up 46 chromosomes. When they unite, it's now known as a zygote. And it stays as a zygote for about 4 days. The cell will divide during those 4 days, going from 1 cell to 32 cells. So 1 cell turns to 2 cells, then to 4 cells, to 8 cells, and so forth. And then it turns into this ball of cells, kind of looking like a raspberry. Okay, we call that a marula. That's another course at another time. Okay? But we'll talk about that splitting, what's actually happening with that. It's called mitosis. So a gamete contains half the number of non-sex cells, or sex chromosomes, If there's a full set of 46, we refer to it as diploid. If there's half, and that's easy to memorize, if there's half the number of chromosomes, then we call it haploid. And there's 23. Now this is in a human we're talking about. In an elephant or a bird or whichever, they can have 92 chromosomes or 142 chromosomes. We're talking about in a human that there's 46 chromosomes. They're doing a practical exam. What you're going to have later on. I'll be doing the same thing in the hallway, so I'm not upset that he's doing that. You'll hear the same thing. When we get to the practical laboratory exam, they're doing this thing. Don't forget. Okay, so it contains half the number of chromosomes. Okay? A gamete. What's a gamete? An egg is a gamete. A sperm is a gamete. Each one of them has 23 chromosomes. Okay? Questions about that? Pretty straightforward, right? Okay. Now, we have this deoxyribonucleic acid, DNA. Every human, I don't care who you are, black, white, German, it doesn't matter. You all have the same DNA. Your brother and your sister have the same DNA. Your neighbor has your same DNA. That's what makes us human. They all have the same DNA. Well then, what makes us different? Okay, it's the genes, right. You see, everybody here has the gene for blue eye color, and they have the gene for red eye color, and they have the gene for brown eye color, and purple, and all that, whatever it is. Everyone has that. It's just for you, the brown eye color is the one that's turned on. For you, or another person, the blue eye color is turned on. Think of this as all of you have the same textbook. You all have the same information, but you highlighted one sentence that the other book didn't highlight. So your classmate highlighted something different. You see the difference? You all have the same information, but at the end of the semester, when I look at it, it's all going to look, maybe yours is highlighted in pink or blue in certain things, and other people did it yellow and orange. But it's the same information. It's just certain things are highlighted. Is that clear about that? All right? But all humans have the same DNA. All right? So DNA holds together each chromosome, and as I mentioned, everyone has it. But that's what makes us unique. Because the blue eye color is turned on for you, whereas your friend has the brown eye color that's turned on for them, but their blue eye color is turned off. Okay? Is that clear about that? All right. So that brings up chimpanzees. That's our closest relative, meaning our DNA is what other species out there is closest to our DNA, the chimp. But as much as we look totally different than chimpanzees, the only thing different about our DNA is 2%. That's it. So we're 98% chimp, if you think about it. Right? The only thing different is 2%. 2% is a huge, huge difference when you look at us and chimpanzees. Do you see what I'm saying? You might think it's only 2%. 2% is a big thing when we're dealing with DNA. Okay? Questions about that? I think the next thing that we're closest to is an orangutan. And the third one, I looked this up, is, believe it or not, a pig. So it's interesting. Yeah. So some people are closer to a pig than me than others, right? So, but anyway, so that's what it comes down to, okay? Do you understand the concept of what DNA is? And everybody has the same DNA, just different highlights. But we all have the same DNA. What makes us different from other species is the DNA changes. Not the highlights. I'm talking about the actual DNA. Okay. So a gene is a trait, okay? A certain segment of the DNA makes a protein. You get the encyclopedia set. Each article about a roach or about, I don't know why I'm fixated about roaches, but all right. But a roach or let's say an alligator or an aardvark, that's a gene. And you could highlight that, okay? So this determines enzyme activity. So if we don't look at it like an encyclopedia set, and think of it an encyclopedia set for all the best recipes for making everything, brownies, cakes, steaks, anything. But you're not allowed to take that encyclopedia set outside the library. It's a reference book. That DNA cannot leave the library. But you can make a copy of that recipe. Go to a Xerox machine, make a copy of that recipe, and then you take that recipe that you just made a copy of, you close the book, put it back into the library, but you take that copy and you bring it outside the nucleus. Took me a while to come up with that analogy. That process of Xeroxing is called transcription. What you're taking out is called mRNA. And we'll talk about that when we get into protein synthesis and make it a brownie. But that encyclopedia set stays in the nucleus, does not move out. But a copy, you're not making a copy of the whole encyclopedia set, you're making a copy of one gene that makes a protein or makes a brownie. That's what's going on here. Does that make sense? All right? So this determines enzyme activity, determines color of hair, skin, body statures, all these different things. Also, the possibility of if you're going to get a disease or not, diabetes, high blood pressure, whatnot. Okay? A genome, as I explained, is the whole thing, and knowing where everything is on the encyclopedia set. It's all the genes of one organism. Humans, it's about 20 to 25,000 genes on the whole encyclopedia set, not on just one volume or one chromosome. We're talking about all 46. This is an interesting thing. Of all that stuff, we realize now, I mean, things can change later on, but we now know that of those 20 to 25,000 genes, only 2% is actually going to be a part of genes. Only 2%. That means the other 98% is stuff that's non-coding. It's nothing to do with genes. Plays a role in more of the structure of the chromosome as opposed to gonna translate into some sort of gene. We kind of call it the junk DNA, because really, it is DNA, but it's not going to tell you what kind of protein or brownie. But perhaps we don't know everything about it. Maybe that 2% is really 5%, because they're still doing studies. So we don't know if it's just junk DNA or structured DNA, or we just don't know if it has a function just yet. Okay? Yeah. Oh, yes, you're absolutely right. Okay, I see what you're saying. So there's actually this, so the 20 to 25,000 genes is what's incorporated in the whole DNA. So in other words, if you look at the whole DNA, the 2% of the DNA makes up 20 to 25,000 genes. Is that a good question? Yes. Good question. I'll say it one more time so it's all clear. This has a lot of letters to the DNA. Huge, okay? Of this huge DNA that's happening, only this much is actually going to code for genes, okay? Which makes up about 20 to 25,000 genes that it makes, okay? Which is 2% of the whole molecule of DNA. Is that clear? Good question, good question. Actually, I should tweak that up a little bit so you can see it, you understand it better. Does that make sense? Is that clear? I'm saying it right, right?
So also 98% they don't really know all that much.
Right. Yeah, the structural things. If we go back to my analogy with the encyclopedia set, okay? The actual words is genes. But where are you going to put the words on? Paper. The binding. If you weigh that, most of the weight is going to be the paper and the binding and the hardcover book. Does that make it the hardcover? And most of it is like 98% the weight. But the actual words, the ink, might be the 2%. It's actually going to code for something. But you've got to have the codes on, you've got to have those genes hooked up to something so that it has a place to go. So 98% is structurally, it's the chromosome structure itself, whereas the actual words, the things that you're gonna, that are going to make sense to you, that are going to code for something, that's a 2% that's going to make the genes. Is that a good analogy? Okay? Right, come to my class, I did these analogies, right? Instead of just memorizing it, I need you to, the analogies, it takes longer for me to come up with them, but I do want you to see, I want you guys to come up with your own analogies. I'm trying to, as I told you before, wire your brain to come up with these things to make sense of it. You know where I'm going when we're going to talk about protein synthesis. You bet your darn bottom dollar that I'm going to be talking about making a brownie, because it makes sense and it gets us hungry. Okay? So questions about this? All right, good question. Good question. So that's what our DNA molecule looks like. It looks like a ladder that's twisted. All right? So we have one side that we call it a helix, which is a twisted one side, but there's another side to it that's also twisted and bound to it. So we have a double helix. All right? Think of helix like helicopter, right? All right, heredity. All right? A trait or gene that's passed down from parent to offspring, generation after generation after generation. A gene or a chromosome can be mutated and cause disease. All right? The disease may not appear until later on in life. You may carry the gene for diabetes, but you're not born with diabetes, but when you reach 40 years old, you had the gene for it and it turned on. So it might not be there right away. It may. You know, we talk about Tay-Sachs disease or sickle cell anemia. Well, those could happen right away when you're born. But sometimes, like schizophrenia and stuff, it doesn't happen until later on. So these are just some of the different diseases. Okay? All right. So, and the other thing too, if there's a hereditary defect that you know that you have, then it's going to help you to figure out what to do with your health later on. In other words, if you know your mother and your father have diabetes, there's a pretty darn good chance you carry the gene also. So then it's up to you to try and decrease your risk of getting it. You probably will get it. I won't be lying to you. You probably will. But maybe your body is programmed that you're going to get it at 40 years old. However, if you eat differently, if you exercise differently to the better of yourself, then maybe instead of getting diabetes at 40 years old, you're going to get it at 60 years old. So knowing about that you have these risk factors helps you determine how you're going to take on your life. Okay? All right, so the chromosome structure. How's this all together? Well, DNA, like I said to you, can be coiled or it could be loose. And the best way I gave you this analogy to make it quick is that you have this encyclopedia set, and if they're all bound into 46 volumes, they're easier to manage. You could separate them. You take 10 volumes, I'll take 10 volumes. They're easy to carry from one place to another. They're bound together. But it's not good to actually try and find where the brownie recipe is. It's easier to take out each page, lay it out very thinly across the floor of that whole entire 46-volume encyclopedia set, and now you just scan and say, where's the brownie, brownie, brownie? There's the brownie. I see the brownie recipe right there. So most times, and this is where I think it's a big misnomer, when people think of DNA, they think of the double helix, and then everyone says chromosomes, and you see these X's and stuff. Most of the time, your DNA is not in chromosome format. It's there maybe 10% of the time. It's only there when you're going through mitosis. That's it. Most of the time, it's thin and uncoiled, and it is laid out like that because your body is making more proteins than it is dividing into cells. Does that make sense? All right. And this DNA, you can see the ladder over here. All right, here's our double helix. It eventually will wrap itself around these histones. And they're molecules over there. All right, they're protein molecules. They help to organize it a little bit better. Okay? Lupus. You've heard of lupus, right? Lupus is actually people who have lupus, they kind of attack these histones in certain cells, and that's what that's all about. It kind of unravels it. And that's why, if you know anyone, or you, who has, there's some celebrities. I know Nick Cannon has it. And there's a few people out there, but that's why they have a lot of different scenarios of lupus. Whether it's a skin lupus, they have this. Rash. You know singer Seal? Is that his name, Seal? He has lupus. And he has that skin that goes like this. Looks like a butterfly with rash on there. Other people, the lupus, they get kidney disease. There's a lot of different things that can happen with lupus. And it's because it depends on which histones and which cells are being affected. Okay? So that's what's going on with that. All right, chromatin, as I mentioned before, most of the time their DNA is put into chromatin format. All right, it's loosely coiled, or not even coiled, it's just loose. Okay, it's uncoiled. The only time we have any chromosomes is when we're ready to divide one cell into two. Okay, and they're in pairs of chromatids. All right, and there's a centromere. So I know a lot of people like to draw chromosomes like it's in X. That's good, you want to draw it that way. But that's not really what it is. In other words, this is not one chromatin. It's kind of this way. So in other words, this is a chromatid. All right? So you'll learn about these in another class. You've got a short arm and a long arm. But that's one chromatid that are connected to a centromere. And then you'll have another centromere here with another chromatid over here. So this was something you originally got from mom, this one you got from dad. All right? Does that make sense? All right? It's not, people look at it as an X, but it's really, this is one and that is one. Not 1, 1. Okay? Just so you can understand the structure a bit. All right? I don't want to spend too much time on that. We've seen that before. and they're trying to show you over here. So here's the chromosomes, right? That's a chromatid on one side, there's a chromatid on the other side, and then the centromeres are in the center there. Okay? All right, so double helix of the DNA. So what it's set up as is that we have a nucleotide, right? That's what they is. It's a nucleic acid, as the name implies. And it's a long chain of thousands and thousands, probably millions and millions of nucleotides. And a nucleotide, just to refresh your memory, is made up of a sugar, a phosphate, and some sort of nitrogenous base. Okay? And the double helix is made up of these two long chains. So, what we have here is sugar, 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 phosphate, phosphate, phosphate. Phosphate, 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 phosphate. All right? That's one side. That's a helix, and it's kind of twists. But we have another one on the other side. These are phosphates. And this is a sugar. Okay? So the backbone of this DNA molecule is made up of sugars and phosphates. Okay? Now, the rungs, that's the sugar and phosphate, all right? Then we have the rungs. Now, what's a rung, right? Yeah, that made up of, right? Yeah, MUO is made up of. Now, that's the backbone. And I didn't realize this about 2 years ago that most students don't know what a rung is. It's the part you put your feet on, the rungs. Okay? There's the backbone of the ladder. When you climb the ladder, when you put your feet and hands, those are the rungs. Okay? Took me a while on that one, but did some tutoring. I'm like, you didn't know this, and I talked to more students, they didn't know it either. Yeah. Not saying you know, I'm just saying we're working on the same page here. Okay? Now they have bases. Okay? So this is a sugar, 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 sugar. And then what's the name of that sugar? Deoxyribose, right? And this will be the phosphate. Okay? The rungs are attached to the sugar, and there are different things over here. And there's one over here too. Okay? So these are our bases. And as you've seen before in other biology classes, the bases can be adenine, thymine, cytosine, and guanine. Right? You've seen the C's and the A's and all that kind of stuff. Adenine will always go with thymine. Guanosine will always go with cytosine. Alright, so adenine binds with thymine, and cytosine will bind with guanine. And vice versa, right? If I'm saying adenine binds with thymine, the thymine will bind to adenine, right? That's probably where it stops for biology, where the structure is. But then we throw in other words. Hydrogen bonds. Oh, we can't get away from these hydrogen bonds. So we have hydrogen bonds that are in between these bases. That's what's holding them together, is hydrogen bonds. And when we're dealing with say this is A, this is T, this is G, this is C. I'm sorry, it's a little bit small over here. That's a C. No, that's a C. So that'll be a T, and this is an A, this is a C, and this is a G. So what's happening over here is that we have hydrogen bonds that are holding them together. There's no hydrogen bonds, there'll just be two separate things. You're not going to be able to climb on it. So between the A and the T, there's two hydrogen bonds there. Wait, is that right? Yeah, okay. And guanine, there's going to be three. Guanine and cytosine. Here it'll be two, and here it'll be three. Okay? Okay? Now, what do we know about hydrogen bonds? Are they strong or are they weak? Weak. Now, look at this. I didn't draw this here, but these are hydrogen bonds. But in between the sugars and the phosphates are covalent bonds. Are covalent bonds strong or weak? Strong. So are we going to be able to break these bonds between the phosphates and the sugars? No. You got to, if you're going to do that, you've got to put a lot of energy to do that. And that's hard to do. Also, the bonds between the sugar and the base, that's also a covalent bond. Are you going to be able to break that easily? No. All right? So these are strong bonds between the sugar and the base and the sugar and the phosphate. However, in between the bases are hydrogen bonds. They're easy to break. Think of this like the zipper that's on your hoodie. Okay? You got two parts of the zipper, right? And they're fused, they're sewn to your two sides of your hoodie, right? It's going to be difficult to break it up. You have to put some energy. You can do it, but you're going to have to unsew it, and it's difficult to do that. But unzipping it, pretty easy to pull apart. All right? You know where this is going, right? We could break up these bases pretty easy. And that's the basis of being able to duplicate this DNA. Now, structure-wise, when we're dealing with bases, sugar and phosphates are covalently bound to each other, right? The bases themselves are either one-ring structures or two-ring structures. And they're referred to as purines and pyrimidines. Purines are our double-ring base structure, and they're going to be the adenine and guanine. And I'll show you pictures of what they look like. The pyrimidines are just a single ring there. And that's going to be our cytosine and thymine. So look what's happening here. We're going to have a double ring, adenine, going to thymine, which is a single ring. You never have two double rings together. You never have two single rings together. My little mnemonic to help you along with this is atomic guns. Right? I got two atomic guns here. Two ring structures. Atomic guns, A and G. Okay? So adenine and guanine, atomic guns, AG. Think of two rings, right? Two rings, atomic guns. And then you say to yourself, well, how am I going to memorize the other one? Don't! Why waste your time on that? If you know that one, then don't worry about the other one. Right? Because then it's everything left over. So that's how it looks, right? I kind of drew it schematically up there, but it's twisted, and the G goes with the C, the T goes with the A. This is a covalent bond, these are covalent bonds, but in between the two bases, they're hydrogen bonds, easy to break. All right? Again, it's trying to show you what I drew up there over here. But here you can see there's 3 hydrogen bonds between the C and the G, or the G and the C, but 2 double bonds in between the adenine and the thymine. Okay? All right, so let's talk about RNA. All right? Basically, it's just one side of that thing, even with a base. That's it. One side of the ladder. Not that ladder, but it's a smaller thing. That's billions and billions of nucleotides together. RNA is usually thousands, which on a molecular level, that's smaller than billions. Okay? But it's only one side, and it's not, meaning it's not a double helix. It's the same as DNA, right? A nucleotide. There's a sugar, there's a phosphate, and there's a base, with 2 differences. Okay? One is that the sugar's different. It's not deoxyribose, but it has that extra oxygen on it, so it's not deoxy, it's just ribose sugar. The other difference is that there is no thiamine in RNA as the base, but instead it's replaced by something called uracil. And uracil is a pyrimidine just the way a thiamine is, a single ring on it. Okay? And uracil will bind with adenine as much as thymine did in the DNA. So these are what they look like. Again, I won't ask you to draw these out, but you should have an idea of that should appear on your exam. But an adenine and a guanine are the double ring ones. See what I mean by double rings? Whereas cytosine and thymine and also uracil that's in RNA, those are all single rings. And there's a nitrogen in there. That's why it's called a nitrogenous base. Okay? So 3 types of RNA. First we have is messenger RNA, or mRNA. That's the thing I was trying to explain to you. When you go to the library, you want to get a recipe in this gigantic encyclopedia set, but you just want a copy of that one recipe, that one gene. So you go and make a copy of it on the Xerox machine, close the book, put it back on the shelf, leave it in the nucleus or the library, and then you take that sheet of Xerox copy of that recipe and you take it out of the library, out of the nucleus. That sheet is called mRNA. It is the recipe to make a certain protein. So it leaves the nucleus, goes into the cytoplasm, travels on the roads in the cytoplasm called endoplasmic reticulum. There's ribosomes on some of them, and that's where the whole process of making the brownie. Transfer RNA, tRNA. That's the place that occurs on the ribosomes. With the recipe of making a brownie, there's going to be things bringing in the ingredients. You need eggs and brownie mix and vanilla extract and water and things like that. Well, those things are attached to something called transfer RNA, and it's going to transfer the amino acid into the bowl that you're making this brownie. Eventually, it'll make a protein. It'll make a brownie. You know where I'm going with it, right? But we'll get into that later. And then we have ribosomal RNA. It's created by the nucleolus, not the nucleus, the nucleolus, and it makes the RNA. It's actually going to help out with, remember how I told you a ribosome has no membrane to it? It's got 2 parts like a clam. You've got an upper part and a lower part, a 40S unit and a 60S unit. Well, basically, the rRNA is going to aid in putting that clam together.